Hi, my name is Jales Fonseca. I am a PhD candidate at the Sorghum Breeding and Genetics program in the Department of Soil and Crop Sciences at Texas A&M University. And today I'll be talking about genotype by environment interactions, adaptability and stability analysis in R. But before I start, I would like to say thanks to JR Biotech Foundation for inviting me to give you guys a talk and participate in this great event called Reach and Teach Science in Africa. It is really a great pleasure to participate and share a little bit of my expertise in data analysis in R. And with that, I want to introduce my topic by presenting a general plant breeding pipeline program, just a scheme of a breeding pipeline. And when we do that, we can actually include some of the molecular techniques that you guys are learning throughout this workshop. And that's why I was thinking about how we could integrate, we could combine some of the expertise that you guys are putting a lot of efforts to learn. For example, extracting DNA, run gels, maybe analyzing a GWAS, having a GWAS analysis, sorry, or having a QTL analysis. So how we would combine all of these concepts that might seem a little different, that has no connection. So if you put this within the breeding program pipeline, you will see the value and where you can apply those technologies. So for example, to start a breeding program, you need to create genetic variability, right? You need to find some genotypes to start crossing them and try to start selecting, find the best, and then release. This is what we're looking here. So you create your genetic variability, and then you select those best ones, and you put those best ones to identify statistically how those best ones, who are those best ones. And once we find them, we release, right? So in this context, the molecular techniques that you guys are learning are extremely helpful to help breeders, for example, to perform GWAS analysis and find some subpopulations within the breeding population. Or even more important for breeders, you can start making QTL analysis, and let's say you're looking for introgressing a very specific trait in this exotic line. For example, you want to introgress that particular gene that controls for a disease resistance. So you will run a QTL analysis, and that will help breeders, for example, to bring and introgress that via backcross, or you can develop a GM trait. That's how you introgress a specific trait into a breeding population, for example. So all of these molecular techniques are really important within the context of a breeding pipeline. But my topic today is related to genotype by environment. So after let's say, introgressing that important QTL into the breeding population, we need to assess those high-performing genotypes in a multi-environment trial, so we can define which ones we should release as a commercial varieties, or commercial to sell as a hybrid or a line, a finished product. And when we do that, when we create those those multi-environment trials, we can establish like a randomized complete block design. We need to have those experimental designs across multiple environments. And when we have that, we start to think about a really important source of variation in plant breeding called genotype by environment interaction. And this G by E is a big challenge for breeders, because we need to see if the hybrids are consistently good across multiple environments. So there are many concepts and many concepts that we need to make sure that we're following, and we need to assess the hybrids. For example, stability. Are those hybrids stable across environments? Are consistently good, or they are only responsive to best environments? So we need to start assessing those. So the focus of my lecture here will be more towards these part of the breeding pipeline. So I hope you guys are interested in this part as well. So, okay, if we're talking about G by E, we need to know how to classify G by E. So first of all, for you to assess G by E, you need to have 2 or more genotypes in 2 or more environments. So in this graph here, we're gonna show all the patterns that we can see once we have multiple genotypes in multiple environments. So when you don't have a G by E in your multi-environment trial, this is what you would expect. So genotype 1 will perform the same way as good or as bad in multiple, in any environment. There is no interaction between those 2 genotypes. But let's say that you have some sort of G by E, but it's just, it doesn't have any crossover on it. So genotype 1, although it's more responsive to this environment, maybe because we have more water available, let's say, this genotype is more responsive to good environments. So it's better than the genotype 2, but it's also better than the genotype 2 in the environment 1, which is different for this case. Here we see that there is interaction, or there is a crossover interaction here. And we see that the genotype 1 is better than genotype 2 only in environment 2. In environment 1, genotype 2 is better. So this is a challenge for breeders, because we need to find those genotypes that are stable and good across all environments. And in this case here, genotype 1 has yields higher in this environment. You see the difference here and here is the same, so there is similar shifts. In here we have different shifts, so it is a different type of crossover interaction. And how can we deal with those G by E interactions? If there is no crossover, so there is no changes in the rank of the genotype, we can just ignore that G by E. Although it is significant, we're going to see how to assess the significance of G by E effects using a linear mixed model. In this case, we're going to learn this today. If we see that the G by E is significant, but there's no crossover, so we just ignore it. Or we can reduce if there is crossover interaction, and we can start creating mega environments to better assess those hybrids. Sometimes the locations are so different that we can't just treat all of them together and run a combined analysis. In those cases, we need to start thinking about developing mega environments so we can better assess hybrids. And then by doing that, we're going to explore the relationship of those environments so we can start assessing the stability of those hybrids within those environments that are similar among each other. And in this case here, it tells you the different types of mega environments and how to deal with the G by E data of if you're assessing yield, for example. So let's say you have a data, and we're going to spend some time to see if the G by E is significant or not, and how to do that in our workshop. So when we start to work on R, it is going to take some time. So it is important. You guys are going to see why it is important. And once we detect the G by E, we need to see if there's crossover. So if there's crossover, we need to start thinking about developing mega environments. And then once we develop those mega environments, we need to assess the stability of hybrids within those mega environments to see if the best performer hybrid is the best performer across all the environments. If that's the case, we're looking at an ideal hybrid or an ideal line, And that would be awesome. We just could release that genotype and be done with it. But sometimes we see that the second best actually perform relatively and stable, and it is stable across environments. So sometimes it yields a little less in the best environment, but it yields significantly more in the low or in the poor environments. So that hybrid would become a more interesting hybrid to release than the one that is very responsive. And we will be analyzing a data for the beginning that is about a grain sorghum multi-environment trial. And after we develop those mega environments, we will be running some stability tests, stability analysis using GGE models and also AMMI models. And some of the stability parameters are listed here. And we will be using many packages, but I'll be focusing on the GGE, AMMI stability, and I will introduce a little bit of Metan, which is a really good package, but since it has more functions than the stability and GGE, I'll be covering quickly at the end just to say that there are multiple ways for you to run a GGE biplot. And with that, I think we can start going and working with R. Okay, so again, my name is Jales Fonseca and my email is here. So if something is, if you guys are not understanding or having a problem to run this code, these codes here, just send me an email. I'll do my best to reach out to you and answer your and try to assess your problems. So I'll be assuming that you guys have some background in R. So I won't be going to every single detail of the codes. So for example here, this is how we're gonna input the data to our R. The setWD function just brings the folder where the data that I'll be sending you guys will be located. In my case, it is under desktop Jales Fonseca. So this opens the folder, the R goes to that folder, and then I ask the with this code here, I just go and say, okay, R, there is a file called lecture.csv within this folder. So read and bring that folder, that table as an object so I can play with it. That's literally what it is written here, believe me. So once you have the data there, you can start thinking about, okay, let's see what is the data about. With this function here, head, you can see, okay, so this data has hybrids, a list of hybrids. There are reps, there are environments, there's yield data, there's plant height, there's flowering, and there's mega environments. We're gonna get to this column here at the end. But we need to see what is that data about. And this is the function that tells you, okay, so what is the structure of the data? So again, every time that you see this behind of a parenthesis, it is just a function. And R has some functions built in R, but when we use packages, we run and use libraries to increase the amount of functions available in R. So with this structure here, we can see that it is a data frame with 900 observations and 7 variables. All of the variables are listed down here. And there's the variable hybrid. We have this hybrid as a character. But since we're going to be analyzing this in a model, we need this variable as a factor. And this is so as hybrid environment. So this is how we convert, we change character to a factor. So we need to put those as factors. So once we run all of these codes here, you run it again, and then you can come here and see, oh, okay, so now I have hybrids as factors, and I have 100 different levels. So this data has 100 hybrids in 3 environments, and each environment has 3 levels. So this equals to 900 observations. So that's how you get to those 900 observations. 3 times 3 times 100. So in this data set, since we have some NAs and probably most of the data that you guys collect will have some NAs, I'm including here as part of this workshop how to manage your data a little bit. So this is how I assess some of the missing data in my file. So we see that there's 11 NAs in yield, missing data, same 9 plant height, and this has 207. All right, so what we can do, we need to remove DNAs now, and there are many different ways to do it. The one way I like to do it is just by running this package here, tidyverse. And if you guys don't have tidyverse, just delete this, run, and just run library tidyverse. It has a lot of functions for you to clean your data. So what I'm doing here is I'm cleaning. So I'm creating a different object. So data is where that file within that folder is. But now I'm starting to clean the data. So I will put that data and I will create a pipe. So this is a structure called pipe, where I'm telling the package to drop all NAs that are within the column yield. So when I do that, remember it was 11. I create another object with last 11 data points. So now all of the NAs for yield is out. So as an exercise down there, I'm going to ask you to do all of these things that I'm doing for all of the traits and by environment. So I want you guys to do that exercise so you can experience R more and get more familiar with it. That's the whole point of this section. I want you guys to be familiar with R so you can be confident enough to maybe next year be in the university, be the person to give this talk in person next year. Why not, right? All right, so once we have the data clean, we need to start exploring our data. So we're gonna run some exploratory analysis. So in yield here, for example, the minimum yield value is 1 ton per hectare, and the maximum is 9 tons per hectare with a mean of 5.6. So if you want to create a histogram that shows this a little bit and see how the if the data looks kind of a normal distribution, you can come here and run this code here. It says create a histogram of the data clean, which is the one without NAs, go into the column yield, and this $ here tells you go to the column yield and create a histogram. And I'm putting the main, which is the title yield, and the XLab here. You can change those parameters. So I'm just leaving it there, and I'm just talking very briefly just to show you how it works. If you want to change the color, just come here, put red. So you guys can start playing with it after. Okay, so once we see that the data looks okay to me, you can start running the analysis. So be aware that I won't be covering the theory behind of these models. So I would strongly recommend you guys to start reading about linear mixed models and why those estimates are the way they are, because I won't be covering here. My point here is just to show you how to run those models and extract the information out of it. So if you don't have the LME4 package, you should install by removing the # and just run the same thing here. This is the package that provides the linear mixed model functions, and this is the one that provides us an easier way to assess the significance of our effects. So just run them, because I do have them already in my R. I don't need to install them, but I need to run so I can I need to use the function library so I can get access to the functions that are within these packages. So now it is important here too. If you guys want to assess if hybrids are significantly different, and you want to see those letters that separates the mean, you call hybrids as fixed. And for you just to tell the model to call hybrid as fixed, you just put hybrids without this parenthesis 1 bar. If you put parenthesis 1 bar, you're going to be telling the model that this effect here is random. So here, what we're doing a combined analysis already, and we are using yield as our dependent variable, as our y, and here is the function that runs the model using that is within this package here. So see all the effects on our model. So we have hybrids, we have environments, and we have G by E, and this is actually what we're interested in. But before we get to the point that we analyze G by E, we need to do all this. So that's why I'm covering all of this also. And this is because probably every single time that you get a multi-environment trial, you should do this kind of analysis to see if the hybrids present in your multi-environment trials are significant. So here is environment, G by E. This is rep nested within environment, and this is how you call what kind of data you're working on. In this case, we're using the data clean. So data equals the data we are working on. So you run this. Now, all of the theory behind linear mixed models was applied, and now we can just see the significance of the effects. And we're using a wall test because a wall test is a recommended test for fixed effects in a linear mixed model framework. A lot of words there, but my point is we're using a ANOVA type 3 sum of squares just to tell the hybrids, just to assess hybrids the right way. I'm sorry for not covering the theory, but it would take a lot of time. And maybe you guys are not even interested. You just want to see how to run the analysis here. So just keep moving forward. Again, since we are interested in the means of hybrids, we might be trying to separate them statistically, correct? So this is how we would do. So for example, we are calculating the blues. Blues is just the adjusted means of an effect that is assumed as fixed in a linear mixed model framework. So these values here in front of those hybrids are the adjusted means of hybrids within a combined analysis. Again, remember, we started doing a combined analysis, and that's not the right way to do it, but I will tell it after as an exercise for you guys to start doing it by environment first. So this means separation is going to take a lot of our time. So I am not going to run now because it is going to take some time, and it's just gonna give you that mean separation test with the letters in front of it. There are some disagreements, so people are not using those compact letters display no more. It is not recommended. What people are actually doing is using these two functions here where they plot the p-values and they compare p-values. If you guys are more interested in learning about learn about these, just go to the multi-comp review VigNet, or try to and then you assess that information, why people are doing this nowadays. Okay, this is for you to assess the means of hybrids and to make a mean comparison, a mean separation. But now one thing that is extremely important in plant breeding trials is to assess heritability. Do we have enough variation in our trials that we can actually make selections? And this is how we're going to make it. So before, remember, we were having hybrids as fixed because we were concerned about the means separation of those hybrids. But now see that this model here has hybrids as random because we now want to calculate the variance component of hybrids. So we can actually have how much of the variation within this trial is due to the genetic component, and that's how we do it. So same thing, the dependent variable is yield, and I strongly recommend you guys to do this for the other two traits so you get familiar with R. I can't stress this enough. You need to use R so you be familiar and not intimidated with all these letters that sometimes make no sense. So just put yourself in, take some time of your day, just play with it. So once you run, you're gonna, this is how you assess the significance of your models. So you can see that hybrid is highly significant, also environment, and G by E also. And even rep was significant. So you want to see the magnitudes of the variance components. You just run summary and write the object that stores the analysis using this model here. So now you see that hybrids has 0.33 of the variation. So this is the magnitude of the variance component for hybrids. And then this is important for you if you want to calculate heritability, for example. So here I put a note to say that it's important for you to test for normality of the residuals of your model. Because if you don't have a normality of your residuals, maybe sometimes if you don't have normality, your analyses are going to be a little biased. So for example, in our case here, we're running a Shapiro-Wilk test and our p-value is low. And as I said here, remember, if p-value is low, nu must go. So what is the null hypothesis in this case? So the null hypothesis is that the residuals are normal. So if nu must go because it is a low, so this is actually not normally distributed. So there is something wrong with this data. And we're going to see that after we break down the data into mega environments, this is corrected. And then it is a way for us to assess genetic information in a better way. So again, and if you want to see the QQ norm plot for the residuals, if this was normally distributed, you would be able to see a nice straight line here coming on the center of this plot. So these are just some diagnostic of your model. It is important. This is good information for you guys to have. Okay, so once we have all of those estimates, we can calculate heritability. And instead of going here, so remember, we can just go here and get this number here, and use the formula of heritability or repeatability and calculate that. But there are some easier ways to use it, and that's how I use. So if you want to go and grab those values, so here, for example, remember 0.33 is exactly that value for hybrids here. So this is how you tell this in a code way. Using codes, you can just create an object that would include that value here. And this makes your life easier because sometimes you have a lot of models to analyze. If you have a soft code, you can just run them. Like here, for example, number of environments, you have 3. Number of reps, you have 3 as well in this case. So let's say you're using a different data set. But because we do have soft codes here, it is always gonna find the number of reps it has and just put it here. So the formulas to calculate heritability or coefficients of variation is all here. So you just run them again. And here you will have, on print function, you will have the mean, the genetic variation, the G by E variation. So these are the variance components. These are the variance components of the models that we calculated. And this is the heritability. This is the coefficients of variation. So you can see the quality of your experiment and all that. So as I mentioned before, there are some exercises I propose we are not going to do here during class, but I strongly recommend you guys to just play with it. So first, I want you guys to do the analysis by environment. Remember, we started with a combined analysis where G by E was included in the model. But I want you guys to do this as an exercise. And then do this for all the traits. The plant height, the yield was done for the combined analysis, but there's also plant height and day-to-day thesis. So once you do that, you can start compare the outcomes. And you're going to see that some hybrids are going to be good in some environments. Some hybrids are not going to be that good for other environments. And then you can compare the outcomes. So across locations and combine, you can see the differences. And then you need to answer, you should answer this. Does the heritability going to increase or decrease for each environment or across environments? You can start talking about it. And there are many ways for you to partition your data. So if you're analyzing by environment, you don't need actually to create another Excel sheet. So to just have that environment. So I put this as an additional topic here, how to sort data in R. So for example, if you want to do a by environment, you will use these codes here. And then you're going to get, so these codes, dplyr is a good package for you to clean your data or for you to filter your data. There are many functions within dplyr. So in this case here, we are partitioning the data. So we are creating an object, a new object saying data clean 1. And we're using the data, the original file that we included in R. And we're creating again a pipe here. And we're saying that within this data, filter environment and just grab the environment 1. And once you run this, you're going to see that this data clean here, it is going to be 300 observations. Let's see if it is going to work. So every time I need to, if that problem shows up, just reinstall dplyr. Sometimes it does that problem. Nope. So you can just come here and finally run these codes. It is going to work. See, I didn't change anything. It ran. And see on this data clean 1 here, it now only have 300 observations. And that's what we want, right? Only one environment. And once we do that, the models that we used before, it is going to change because now we don't have G by E. Of course, there is only one environment. But I want you guys to experience this. So do for yield, plant height, days to thesis, environment 1. And if you want to change the environment, just put environment 2, then environment 3, right? As an exercise. So up until now, we were just making sure that our data is looking good, and we see that there's G by E. Now we need to assess that G by E. And finally, now we're going to actually do a GGE by plot analysis and develop those beautiful graphs that it is common to see in the literature. So for those, I'm going to use a GGE package and an agridata package. If you don't have, just take the # out, delete, run, and then run the library for those 2 packages. So since I'm going to use this GGE, there are some parameters within the package that you need to be familiar with. So if you want to, there are many ways for you to center your data. If you want to create a GGE by plot, you should center in an environment. You can center in the genotype, but you can do that. But if you want to do a GGE with environment centered, just on the parameter center, just put equals true. If you want to scale your environments, just put it true. If you want to see a which one where polygon, just put whole equals true. It is going to make more sense when we start using the packages. So here, what we're doing is we're creating an object that is going to develop a GGE analysis. Again, I'm sorry, I'm not going to be covering the theory behind of the GGE analysis, but this is going to include, so once you run this, all the singular value decomposition is going to be created. It is going to be calculated, and then the variation is going to be maximized in using vectors. So all of that theory should be covered in the literature. So you can just Google the literature there. So Dr. Yan and collaborators, they have a lot of papers published on it, and I recommend you guys to read to make sure that you know what's going on. So we have the object with the GGE analysis done. So the way you run this, you need to have the data that we're working on, which is the yield data cleaned, right? And here is how you include the GGE parameter. So yield will be a G and then asterisks environment. So this is how you put the data to work. So once we have the model here, we can just plot GGE. So this is a diagnostic plot of the GGE analysis. And we can check this with details. So here tells you this is a discrete plot, tells you how much of the variation is explained with the first principal component, the second, and the third principal component. Here is just a mosaic of the environment 1. So what is the performance of the hybrids in environment 1, environment 2, and environment 3? See that these hybrids here are low performers in environment 1, but some of them are good performers in environment 2, and also kind of a little better than environment 1 here in environment 3. So this hybrid here was really bad in environment 3, but good here. So remember that crossover interaction I was talking in the beginning. So this is what's going on here, and this is a mosaic also of the percent of variation explained on the first component. So here, most of the variation is explained by G, and the other two is explained by the G by E. So that's a diagnostic plot. So now we're going to create the GGE by plot. So to use the GGE, the biplot function, you need to have the object with the data analysis, with the GGE analysis. Here, I just created a title. This creates a title here, so you can have whatever name you want. This parameter here changes the size of the font for a genotype, change the size of the font for environment. Here, you can just use different colors, yellow, for example, if you want to have that yellow. So see, it changes the color here. And you can put different colors, so you can play around with it. And whole again adds the which one where polygon. And when we come here, I select a terrible color here, by the way, but all of these yellows are the hybrids. These reds, these vectors here, are the display the environment. So this is the environment 1, environment 3, and environment 2, environment 3, and environment 1. See that there's one, so this is the which one where polygon. So you see that there is a winner in environment 1, which is different than environment 2 and 3. So this area here indicates that these two environments should be treated as a mega environment. So these two environments can cluster together, and this is a little bit different. So that's why we need to investigate G by E. All right, so we know that there is two mega environments, one with two environments and the other one with one environment. So now we need to see that in a better way. So let's just change this to a different color. Let's put it blue again, because now you can actually see the hybrids. So now what we're gonna do is, since we see that there's a pattern in our GGE biplot, we're gonna create a GGB biplot, which creates a mega environment. So for this case, again, the same type of data, same model, but now we're gonna cluster our environments based on the ME. Remember that last column, that was a ME. So this is for mega environment. And I knew already beforehand that that was the way the data would perform. So I created, after I analyzed all this, I went back and I created that column calling mega environment. And then to create the genotype by block of environments, you need to add this parameter there. Once we have the model that creates the mega environments, and same thing as previous, but now look what we have. Here, now we have those two environments clustered together. It's a better way to see the data. So it's just a prettier way to see it. The percent of variation is still explained the same amount. So it is 66%, the first principal component. The second principal component explains 23%. Both together explain almost 90%. It's a good biplot. We can extract a lot of information from that plot over there. So once we develop these mega environments, now we need to start thinking about, okay, now we need to assess stability. What is the stability of hybrids within mega environment, these mega environment 1 here, for example? So we can go again, use the dplyr as we mentioned before, and before we were just analyzing by environment. But now, since we know that there are two environments defining a mega environment, so you can, oh no, you can just run these codes here, and you're going to create a data clean 2 that would include 600 observations. And these 600 observations are the two locations that we had, right? So again, if you want to assess, now what we're going to do is we're going to assess the stability of those hybrids. Oh, wait. Now here. All right. So this plot calls means versus stability by plot. Is the mean versus stability view of a biplot. So see, now we have a different layout. This vector here, we call it an average coordinate of average coordinate environment, which is the average performance, kind of it divides the mean performance across those two environments. And this tells you the best hybrid for this mega environment. So this projection tells you that this is the best hybrid, but it is a little longer compared to the second one. So the second one is a little bit more stable than this one here, and they don't have many differences. So maybe it would be more interesting to release the second best hybrid than the best hybrid. You see what I'm saying? And in this case here, and we can only do this plot because remember when we saw the diagram explaining variation on the first principal component. Let's go back there to just see here. In this case, the first principal component is primarily explained by genetic effects. And because we have that, we can plot that mean versus stability view of a biplot. Since there are many hybrids involved in that data set, I am just putting here a different data set with less hybrids or genotypes, so you guys can see a different plot, and also you guys can play with your own. So here, oh, it is too tiny. There you go. So here you see the same scheme. We have the environments here. All the hybrids are spread up across. And one thing that is important that I didn't mention before is the closer the angle between these two vectors here, the close related those environments are. So these two environments are more closely related than these and these one here. So those plots are really useful for you to start thinking about how the what's going on with your data, with your multi-environment trial data. As another example here, I included a data from this paper written by Crosa, where we have 18 wheat lines and 25 locations. So it is a pretty figure you can create by yourself, and that figure is what is the figure that they published in their study. So you can see here there are 25, was it 25? 25 locations and 18 wheat lines. So the 18 lines are all around, and you see there are 2 mega environments here as well, and you can clearly see when you color them differently. So this is pretty much what I was trying to do, and there are also so now you can guys play with this by your own. So in this case here, what I'm showing is you can create those plots in 3D as well. So it is kind of nice. You can just play with it now. You have the codes. You guys can just start creating your own plots. I included more stability analysis using AMMI, and you guys can just use it. And it is a different analysis now. We're using a different model. Unfortunately, I won't have time to explain the differences between the GGE and the AMMI analysis. But, oh, this is important. Here, so the AMMI model considers environment when you're creating your singular value decomposition, which is the difference between GGE. And again, you can start also seeing some of those plots here. So in this case here, in this case here, you're going to see that environment 1 and 2 are similar. So the angle between those two are smaller than these two, right? It is the same trend that we saw before, but they are using a different method. And again, this is AMMI 1, oh sorry, AMMI 2, because we have the principal component 1 and principal component 2 here. But if you want to run an AMMI 1 where you have yield in the first principal component, you can also plot it. So these is we can increase this. So you can see how the hybrids performed across the environment. So environment 3 was the best environment, then environment 2, then environment 1, based on this axis here. And here is how the variation present in the first principal component. And you can discriminate the environments pretty well. The hybrids are similar in here, but you can see that they perform very different. So 78 performed more than 7 tons. Remember, there was one, the maximum was almost 9. So this one here and this one here is the lowest, which was probably 1 ton per hectare. And if you want to assess stability based on AMMI models, you can just use these functions. And here, there are many different stability parameters for you to use. I'm just showing you. I'm sorry, I think I'm putting too many information, but too much information, but I think you guys will have a lot of time to go over these packages and to play with it. So it would be interesting to bring a lot of information for y'all to just play with it. So here, what we're looking at, so we have the means, we have the rank based on the means, we have the rank based on the Rix equivalence. So this is the Rix equivalence because we are using all the principal components to decompose the AMMI stability. to calculate the sum of squares. So this is, I'm sorry, again, it's part of the theory. So based on the ranks of the stability analysis, this hybrid here was the more stable one. But if you see the rank based on the yield, it was almost the worst one. So we have 100 hybrids, remember? And he is the bottom 5%, 10%. So we want to have, and if you sum these two ranks, we come up with this value here. So if you sum 94 plus 195, so this value here tells you the sum between the ranks. So the shorter, the smaller is this value, the better. So let's find one here that has a relatively low number. 17. So see, this hybrid here is good because it is the 7th best hybrid and is the 10th more stable hybrid across all of the 100 hybrids. So this could be a hybrid. And then once you identify this hybrid, you go over your trials. Remember that you guys are going to exercise doing by environment. So you go back and you can just check to see if this actually was one of the best across all of the trials. And if you see the correlation between many different stability indexes, you can just create this plot here. So most of the indexes are similar. So different stability index is not changing much of the outcomes of our analysis. And also, since I think I'm over time now, there is this library, this package Metan, and I strongly recommend you guys to read the VigNet of this. It provides you a lot of different functions, but and I summarized some of the functions here. So I'm only focusing on creating GGE using the Metan. So there are many different biplots you can create based on the basic one that we created, the mean versus stability, which one where. So I am just putting some examples here for you guys to see how it works. So I'm using the data from Dr. Grossa, which is the wheat example. And once we see this, you're gonna be able to see pretty much similar of that plot that we see before without the colors. Remember, here was yellow and blue and all the, so it is different colors, but we are not separating by mega environments. It's just a different package to do this. If you want to do a mean versus a stability, again, you can use this one here. This is how, and then the only thing I changed, it was the type. So you can change the type of plot you want based on the scheme that I just organized in a nice way for you guys to just come here and change. And another one, if you want to examine an environment, you can just run this. You see, there are many different plots for you to assess the data that you're playing with. So I think I have talked too much for you guys, and I hope that you guys do the exercises. Again, I'm proposing another one here. Use this Metan package to work on the data that I'm providing you guys. So try to explore R as much as you can. There is only one way to learn R, is by using it. So I encourage you guys to play with it, get familiar with the packages I showed you guys today. And if you're interested in the theory behind of all of those packages, you can start Googling VigNet, which is, I showed before here, the word is VigNet. So here you put VigNet and then put the name of the package in front of it. You're gonna see that there is plenty information, and then you start reading about those packages and you get some of the theory as well. And I just want to leave a final note that there are additional ways to assess G by E and stability. There are other parameters, for example, the Finley-Wilkinson regression that you can use. There are other models you can use as Bayesian models, and there are other analysis. For example, if you're using random effects instead of the models that we used for GGE, so these models are based on fixed effects. But if you want to use a random effect, there is another model out there called factor analytics models. So there are additional ways to analyze G by E. But I guess that I presented, so bottom line here, we learned a lot about data analysis in R, how to extract genetic information from a multi-environment trial, how to create GGE plots, how to do AMMI analysis. And I'm available for any suggestions. If you guys want to, if you guys are having problems to analyze your data, my email is here. Again, just send me an email and I'll do my best to reply. And with that, I hope to meet you guys live in I don't know when we're gonna meet, but I hope to see you guys live, and you guys can also bring your questions. Thank you very much for your time and enjoy the workshop. Bye.